So let's take a look at how Mendel's model works with respect to the central dogma of biology. So all of this has to make sense in terms of, in light of what we know about molecular genetics. So how does something become masked? How does something get carried? So if you see here, what we have now is here's our chromosomes, the gene locus. Okay, so the gene for purple flowers has a segment of DNA that produces a functional enzyme that helps to synthesize a purple pigment. Therefore, this piece of DNA is expressed now through RNA and producing a protein. That protein produces a purple pigment. Okay? And only one allele is necessary to do that. So the second allele, produces a non-functional enzyme, or doesn't produce an enzyme at all, perhaps. Okay? This sequence of DNA is different. It does not produce that which can help to synthesize a purple pigment. Lack of pigment in this case would be a white flower. So how do we take Mendel's work back to central dogma? A dominant trait produces a protein that's functional, that produces a protein that helps to express a trait. A recessive trait does not produce that same protein, and therefore the trait expressed would be different. So this individual seen here would be carrying on the red chromosome here, would be carrying the allele for the white flower. So we know that our genes on our chromosomes is our genotype that carries the information for or codes for the phenotype. In this case, genotype being some sort of brown rat or mole, whatever that little dude is. We can be either homozygous, having both dominant alleles. Homozygous recessive could be having both recessive alleles, or we can be heterozygous. Hetero as in different allele types on those individual chromosomes. So now notice how that affects the gametes that can be produced. So in the top homozygous parent, the only gametes that can be produced have the dominant allele. The bottom heterozygous parent, half the gametes will be dominant allele and half the gametes will be the recessive allele. So that takes us to the Punnett square concept. Okay? And the Punnett square must be able to predict Mendel's data, must be able to predict Mendel's 3-to-1 ratio. First of all, a punnet is a small wooden box that was used to separate produce. And so it was a very natural term for Mendel to use, being the overseer of the monastery garden, that I'm sure these kinds of separating boxes were used regularly. And how convenient are they? They make it easy to separate produce. They make it easy to buy one entire punnet at a time. Handy and dandy. Certainly would have been very much a commonplace in Mendel's world in the monastery garden. Okay, so thus the Punnett square concept. Okay, let's take a look at the monohybrid, mono as in following one trait at a time. So here is our Punnett square for one trait. We show the separation of alleles, or what Mendel would have called factors. And here are your 4 punnets for this Punnett square. Notice the offspring predicted in this Punnett square demonstrate Mendel's 3-to-1 ratio. So the monohybrid cross for one gene has to show the same data that Mendel was producing in the garden.